Bache Himanshu, I did actually. First of all, welcome everyone for the today's class. I hope you're all doing well. My voice is coming fine, and you can see the picture. So today I went to this software. I did this. Oops! No, 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 no. Yeah, I did. I tried to uh, make this resolution to 14490. It was earlier 1240. But it didn't change the output actually, and time was running, so I want to share my link today, but I will try tomorrow if I could increase it. But don't you don't all have to worry about the making it to 480p's 720p, because when we gonna go through the presentations, they will be in bold letters, big pictures, so you might not get hindered because of that. Just make your landscape view over if you're going through your mobile, or if you're on the laptop. Yeah, laptop is like big screen, there will be for sure no problem, so don't worry. So any questions you have from the yesterday, anything that you want to ask, I will be happy to answer those questions. Anything that you have, any questions or anything, I will be very happy to answer them. So I was answering to some of the students questions about review articles, that it should be minimum 25 pages, and um, yeah, it should not include your references in that. I'm saying that because once you have a good amount of uh, good quality work, it should not be just quantity, there should be also quality, has to be maintained. It should not be harnessed and maintained properly while you're writing it. So please take care of that. Okay, good. Today I got my vaccination done in Delhi. It's finally done, so tomorrow my parents will get it done. But they say that in Germany they had this Pfizer vaccinations. Many people are getting it, but they are getting ill effect of that vaccine. So we don't know what is the conspiracy theory behind all this. What's going on? And they are saying this is third world order. This is beyond this topic of our today. That there is a thing that in the world they want only people who could survive the pandemic, and the one who will left will be chosen for some task. So we know, like the futuristic movies that we see, like Matrix and all those sci-fi fiction. So we are somehow part of that thing at the moment on a very big level. So let's see. I don't know. I can't comment on these things. So many rumors are coming every day. Um, so till we have our last breath, we will learn and we teach. That's I could say. Okay, so where we have to start today? We have to start from our basics of molecular biology. बच्चे थिंग इज जो आपका जो टॉपिक है मॉलिक्यूलर बायोलॉजी एंड बायोकेमिस्ट्री टेक्निक्स, ये अपने आप में ही बहुत वो है, क्या कह सकते हैं, इंटरेस्टिंग भी है और वास्ट भी है। क्योंकि आप कोई भी टॉपिक, कोई भी आप रिसर्च उठाएंगे और आपको उसके लिए रिलेटेड कोई एक्सपेरिमेंट करना है, या आपको कुछ थिंग्स को आप नोन करना चाहते हैं, सो उस सेंस के अंदर, उस वे में आपकी कितनी ही टेक्निक्स हैं मॉलिक्यूलर बायोलॉजी की और बायोकेमिस्ट्री की, जो कि काफ़ी मायनों में आपकी काफ़ी हेल्प करती हैं। सो within the molecular biology and your biochemistry techniques, each techniques, when you start your experiments, they themselves, each one of them play a quite important roles. You know, it's like it's not possible we can even think of doing research without these two techniques. So they are the mainly making a basic foundation. It's not only for the bioinformatics students or genetic students. Actually, these two techniques will be used by all the geneticists also, RT-PCR, normal PCR, your Western blotting, DNA microarray. So all the light field scientists, all the researchers at some point, proteomic specialist, they need the help of a molecular biologist or a biochemist. Without their help, it's not possible to get into the results. So what we are gonna study itself has very importance for your upcoming career, and on behalf of that, this will shape up your future. Like, if you're gonna use this certificate, sorry, from the IIT Kharagpur, this will really value your CV. Beyond that, you can use it for the various interviews that you have a practical virtual experience, because this is online time, and it's very hard time, COVID time. It's very hard to give very practical knowledge about everything. So in that sense, you will learn a lot of techniques during this time. And so basically, we will go through the basics principles of the most of the techniques, including your MBBT, your molecular biology and biochemistry, and we will also see how we do them in the lab practically. So we will have a lot of videos also for regarding the same. So that's how we will run for our workshop. Yeah. Yeah, sorry, that's one thing. Can Manga? Can I say your name, Manga? Manga, maybe. Um, yeah, I will keep this in English. I can sorry for that. I just switch myself for the sake of some Hindi students. Maybe they are present. But I will keep my language English from now onwards. Okay. Anjali Mahole, please ask in the group. Like, I have shared some topics yesterday, and you can group together and start for that. You can watch the yesterday's video also, it's available. So, everything has been explained on that. Okay, cool. So let's start today, our first day of our workshop. So first 2 days we had our introduction, and second day we have learned like how we will run with the things, how things will run from the review article to assignments. So now gonna start with cell. This is very basics, class 12th. I brought the very basic slides here for the class from the class 12th or 11th one. Maybe in 11th you have cell of plant and animal cells, this chapter in ICS board and CBSC board. Yeah. So here we can see that um, on the left side is the factory, on the right hand side uh, is the cell. Yeah. So both analogies, we will discuss how in a factory things works and how the same way things work in the cell. So how can cell be like factory? So in a factory, we know we have many places, like we have boundary, security guard, CEO, janitor, movers, and so all people working together to make to make the products working, to get things going on, whether it's a Coca-Cola factory, whether it's a Britannia Mary Cake factory, or anything. So it's all everyone's effort together, coming into together into that part, and making things done for everyone. So that's how the cells could also be act like a factory. So we're gonna understand this thing in this upcoming sessions, how it could be. So the building which supports shelters, workers, build goods, maintenance, crews, cleanups, messes. So all these belong to this part. So a cell can be thought of as a factory with different departments, each performing specialized tasks. So first thing is first, cell wall. So cell wall acts like a security gate. So a security gate protects the factory and helps keep it safe. So all the cell wall around the cell, which is the first layer, It keeps a very nice boundary said that any extra material should not be entered. Anything unwanted particles should not make your cell wall of your plant make destructive. So this is how cell wall works. Then comes your cell membrane, which is beyond this main entrance. It's like your office, main office entrance, front doors of the office. So this also regulates what should be entered, what should not be entered from the building. And the cell membrane does the same thing for your plants and animal cells in this case. So who should come in? Is there any interviewer or any salespersons or some students or anyone according to the factory? It will the same way cell membrane will work. Then comes, I know students, participants, you might be someone very seniors in this. You know all these things, but the thing is, I want to teach these things in order to just refresh our memories from our cell biology classes we have in the first semester or second semesters, and to bring them into very simple and in a crisp way. Yeah, not too much details here. So the nucleus, it's a big boss. It's like a CEO, which controls everything of the building, like dean you can say in a college, or the factory manager, who controls what should be come in, what should goes out, how much production we need, how much production we don't need, on which day we get off, on which day we don't get off. So all these things are being decided by the CEO. That is your nucleus. So nucleus work for same your plant and animal cells, and is a big boss. So whatever the nucleus is, we have to follow it. Then cytoplasm. So factory floor has the all workers, machines, equipments on it. So cytoplasm is like in the cell, it's all empty place. All the organelles, your membrane bound organelles, non-membrane bound organelles, they are residing within the cytoplasm. So it acts like a floor of a factory where all your workers, machine, equipments, they are working on it. So it has all the organelles of the plant and animal cells over the cytoplasm. Then comes your endoplasmic reticulum, ER. This is basically an assembly line where workers are standing to do their work, and there's a conveyor belt on that, and things are coming like your any Coca-Cola factory where the bottles are being filled automatically and then assembled there, getting the on the top their cap, and then going for the packing. So this is all done within the factory on the conveyor belt with different parts of the plant. But this same things could be done with the help of ER in the cell in order to transfer your nucleus, your mRNA from your nucleus to the cytoplasm. So in this case, your rough endoplasmic reticulum, smooth endoplasmic reticulums are playing role. So this is the conveyor belt we can see. So things are working on it, and all other are helping to make things done over. Then comes ribosomes. Ribosomes are like workers. So assembly line was the your ER actually. This is assembly line is mainly your in the center is your ER, but the workers who are working on the side chain, who are thinking that how many plates are going on, how many boxes are moving. So these things are being taken care by your ribosomes. So ribosomes are like workers in the assembly line. So they are responsible for building the goods in a factory. Like how much do we need? And is there any error going on? Then we should remove those particles from there. So in that sense, it's helping a lot. So they actually ribosomes, as we know, there are ribosomal units, 70s ribosomal unit, 80s ribosomal unit, and they mainly help to build your protein from mRNA, which is coming out of your nucleus. So from nucleus, your mRNA is coming out, and it's helping to build them into translating them into the protein with the help of your transfer RNA. So we will discuss about basically the central dogma. I hope you know all about the central dogma of life, which means production of your protein with the help of first transcription from DNA to mRNA, and then from mRNA to protein with the help of translation process. And this whole process coming together is known as your central dogma. and which is central to the life, which makes things working out in our cell, and which makes our cells working on, we are having a good life at the moment. So central dogma, that's what the name has been given, central dogma. Yeah. Then comes vacuoles. What are vacuoles? They are like break rooms, like restrooms, where you have a storage. You can put your extra stuff in it, and all the trash materials could be go into that part. Same in the cells. It can hold wastes and stores a lot of water in plants and animal cells. So vacuole role is to hold all the waste and stores in the plant and animal cells. So which one has the large vacuoles? Is it the plant or animal cells? Any comments? I have so many things open, I should have closed them. Bache, is it still hazy? Can you all comments? Bache, how about the others? The presentations that was going on, were they hazy or yeah, plant cell has the highest. So Himanshu, Saksham, Shubham, Anjali, were the presentations fine with you? Is it there's any problem so far? Anyone could please tell me. Ananya Anand. Okay, it's hazy. I don't know. In my last, no one has complained about that. It's yeah. Try to make it the landscape mode. Then it's pretty good. Yeah, because I while I'm playing this presentation, it's also open on my mobile, so I keep checking if how things are working, if there is any problem with it, so I get to know about that. So 480p it's blurry because if I make it to 7, I have 20 mbps, 20 mbps plan. No, sorry, 50 mbps plan in my house. I got the highest plan, but sometimes what happen also that if I make it to 720p, it will streaming just get off. I don't know why. That's why I'm a bit skeptical about 720. Um, yeah, it's still 480. So 4 to 5 students have problem, but rest doesn't have this thing, because if you can't see things properly, it will not make sense for me to continue. But as of now, I hope all the presentations in the PPTs are being fine. There is no problem because it's very big fonts in them. So yes, you're right. We continue. Plants has the highest um, plants has the highest uh, vacuoles, largest vacuoles. And don't worry students, I will share all these ppts with you once I'm done with these lectures. This I always do, whether I'm in college or anywhere if I give my lectures. So whatever the work that I have with me, I love to share with my near and dear ones. I don't like to keep with myself. And yeah, so once the lecture is over, I will share over the telegram group, so don't worry about that. Then comes lysosomes. So they are like your, like in your storage room or anywhere, like in your college also. There are some people who maintain the electrical part, who maintain all the cleaning, who maintain all the mess canteen part. So who person which repairs everything, clean up everything. If something is got broke down and repair it instantly. So that person's the maintenance person, the janitor crew. In US or in German, we call them as janitors. And for all these things, they have only one person actually. He is a multi-purpose person who could fix your electricity thing, who could fix your gas, who could fix your bathroom, who could fix your bed, anything that you say. And for the whole building, they have one person. That's how the rule are. But in India, we are having for each thing a different person and different company for that same. So nonetheless, so lysosomes work the same way. That is all the cleaning part, all the taken care of the cells, that things are going fine for the well-being of the cell. Then comes your power house of the cells, the power plant, that is your mitochondria and chloroplast, which gives energy in the form of ATP, as you might have studied in this biochemistry, playing quite big role. Yeah, so they are the power stations which supplies energy to the factory, and also they are the animal cells, like in the mitochondria, the source, and the chloroplast in the plants. They do for the same in the cells. Yeah. So this is the example now. That is, uh, the whole cells are being shown in this part, where we have Golgi. Like, let's start from this part: membrane, which is controlling outer membrane, cell wall. Then comes cell membrane here. Then after the cell membrane comes your, yeah, endoplasmic reticulum, where your assembling is done. Then this is cytoplasm, this whole area. So things are being transferred from here to there. In the center is your nucleus, the CEO, where your chromosomes, gene production, hereditary units are being taken care. Then the Golgi apparatus is there in collecting the stuff from the nucleus. Then the proteins are being produced with the help of ribosomes on this assembly line. And then in the mitochondria, which is an ATP, like ATP production of the cells, the powerhouse which is giving the energy. So this is a quite nice summary of analogy of your cell towards your factory. So that was very funny way to explain you today, cell and factory, how everything is interlinked with each other, and how can we see the cell from that perspective. So I think we had brush up our memory about cell. Uh, those who was not aware, so you might be those who know about it. Uh, they also got to know about it today. Yeah. So this is not required. This last slide, you can forget that. That was some other students. So that's the slide for the intro, for the cell like a factory. Yeah. Now I want to take you to the journey of uh, basics of molecular biology, very basics, which is being asked also in your various NET exam, your GATE exam, your any post graduation exam you want to do, the MCQs. So from the molecular biology, it has quite good weightage, and they are very simple questions. You can get the things very done easily, but once your concept is clear, things will be fine. Like, how much is the diameter of DNA, which things makes DNA, in proper helical structure, what um, what are the various mutations that happen during the UV light, how can we recover it. So there are many these small questions are being very important from the examination point of view. So we were preparing some MCQs today, and it's just the MCQs is all about, if your concepts are clear, you're clear in crystal from the mind, uh, no one can stop you to get 100%. So this subject this thing is not only to make you standable for from the point of view in the industry level, but from the academic level. If you are preparing for any entrance, it will be very valuable for you. So based on that, let's start this second part. Let me see if there's any questions. So what we will do, I will finish one presentation, then I will come back to the question Q&A, and I will try to my maximum level to answer them, and then we will start with the next phase. So in this way, we will be in touch with each other, and you can ask her any questions here, or otherwise I try. I see some messages coming late night to me till 11 p.m. also. So please, after 10 or something, give me some rest. Because I have a habit, if somebody asks me anything, I need to reply. I can't just leave it like that. Um, so please try to message in the morning if possible, not late night. Yeah, please, somebody back or yeah. Okay, cool. Um, well, but otherwise, I will try my best to reply. There was one question from girl. I don't know. Microbiome was asked. I just said wait, but later on, then I went for just sleep. I couldn't answer that. But today I will try to answer that thing also again after these lectures. Or you can ask if you have a question freshly. Just Viber me. Sorry, Telegram me personally. So I will try to answer on that too. Okay, so that was the Q&A part, how we can discuss things. Okay, let's start with this basics, very basics of your molecular biology. So in this, we just gonna discuss about DNA structure, RNA structure, transcription, translation, various kinds of ribos, RNA, and then genetic code, how the genetic code is made. So you know all the molecular, the 12th class molecular basis of inheritance, that chapter that you had in your class 12th. So we will just discuss basics today about that same. Okay, let's start. First thing first, the difference between eukaryotes and prokaryotes. So whenever you're gonna start with biology, this is the very you can say routine thing that we have to start with. Difference between eukaryotes and prokaryotes. So eukaryotes, they includes plants, animals, fungi, whereas prokaryotes includes your bacteria, you know, cyanobacteria, whereas the cell wall is absent in the eukaryotes. On the other side, in prokaryotes, as present. The exceptional is the plants, they also have the cell walls. And then the ribosome size is around 80s, and the Sydberg unit and prokaryotes at 70s. Whereas the meiosis is happening in eukaryotes, it's not happening in prokaryotes. And beyond that, cell organelles with the membrane bound, they are all present in the eukaryotes. So that's why we are more advanced. That's why we can function. We have this evolution thing in our in the human race. So like nuclear membrane, ER, Golgi apparatus, mitochondria, chloroplast, we are all in the eukaryotes, they are present, but in the prokaryotes, they are all absent. That's why they're primitive and they couldn't develop much. So molecular biology, if we see the basic definition according to the NCBI, so it's a study of molecular underpinnings of the process of replications, transcriptions, and translation of the genetic material. So it will include, first of all, your DNA is there, which could replicate within the cell, and then further it could transcript to produce your mRNA, and then go for translation later on to produce protein. So talking about all these things, all these three different topics, you know, and bringing them for the core studies. So that's what the molecular biology includes, and it's a very interdisciplinary field. If you That's we have discussed today in the first 5 minutes of our lecture, that it has many other techniques which is interlinked with molecular biology and also your biochemistry. Without that, it's not possible to answer things. So it has molecular entomology, like to study your all insects, their habits, their way of functioning, their inner system. And then comes your mathematical biology, includes all mathematical equations in order to solve the biological important questions. Then genomics, that our whole genome is made up of, and out of that whole genome, maybe only 1 to 2% is mainly working for producing the protein, and the rest are just lying there for nothing. So that's how it's also very important. Then structural molecular biology. So once we know the protein structure or enzyme structure, it will be very nice to know its function, where the catalytic subunits are present, where are the, you know, where are the mm, how it could function more better, how it could function better, how can we find an inhibitor for that particular protein in order to find the drug cure for the same, disease cure for the same. So we can find some drug also with the help of structural molecular biology. Then comes enzymology, like it helped the your factory or the Biocon company that came from the Kiran Majumdar. So she made this papayas enzyme in her garage, during her earlier years where on the startup, and from that she just made a big heights, and she's unstoppable at the moment, and a big big businesswoman. I'm very proud, like she's my role model at the moment. Just go through her biography, and she is doing really good at the moment also for the vaccine production in India. Then comes your extremophiles with the bacteria, the microbes, which could be, you know, survive in the extreme conditions, whether it's high temperature, high pH, high salt concentrations, all the extreme these things, and how, what is the reason they could survive, which genes are helping them in order to survive them. So if we extract those genes and try to incorporate into produce into as a drug, so that we can also, if our soldiers who are working in the Siachen glacier or in the Kargil area in the most extreme temperatures, and we can find some microbes who are surviving there too, and we can extract those genes, and we can create one medicines, we can give those capsules genes containing to survive the cold temperature, so our soldiers could also survive there. So DRDO is working on that with the help of some plants at the moment. So also had my 6 months internship at DRDO in Delhi, where we have collected the extract from sea buckthorn. You know, sea buckthorn, it's a plant which grow in the hilly areas of Himalayas. So we just saw that the leaves and the fruits have a lot of antioxidant properties which could help soldiers in order to survive the extreme conditions. So that's how molecular biology could be important there too. Then comes your plant development and defense system. That's very important. Like if we are seeing our immune system, our immunity in the human being, the same way plant has also very typical and complicated, quite complicated system. So in order to do that, this molecular biology is also quite helpful at that part. So all in all, so it's very essential, very important, and has many roles in different um, areas, from cell, plants, microorganisms, molecules, populations, animals. So this summarize this picture that we are seeing at the moment. This actually summarize everything. Now, so some definitions, how the things were discovered, how things were done. So let's see this field, as we have discussed, the area of biology and chemistry. So they belong to this genetics and biochemistry. So they got somehow overlap with each other. So they all are interconnected. And whereas where particular the molecular biology itself, it has interaction between various systems of the cells, like your DNA, RNA, protein biosynthesis, and how these interactions are regulated, how the DNA production is being taken care, how the RNA production has been taken care, and how the protein biosynthesis has been taken care. So all these things, your one, two, three, this is your molecular biology is revolving mainly on these three different parts. So much of the work in the molecular biology is quantitative, and recently much work has been done at the interface of the molecular biology and computer science in bioinformatics and computational biology. So since the late 1950s and 1960s, molecular biologists have learned to characterize, isolate, and manipulate the molecular components of the cells. Organisms include DNA, RNA, and the protein. So we have understood like how we can, even with the help of your restriction enzyme, restrictions endonucleases, we can cut our DNA segment, create sticky ends, and we can add the other segment of plasmid into that and create a new recombinant DNA. So all these things which came out to be later in the 80s and 70s, we have discovered these things for the betterment of the human being. That's the where the biotechnology comes. So using all these your microorganisms, live organisms, for the welfare for the human, is the technology known as biotechnology. So coming to that point, basic component that your molecular biology involves, as we have discussed, that is your DNA, RNA, protein. तो इसी पे हिंदी नहीं, sorry मैं, because in my college everyone understand Hindi, they don't they want things to be in Hindi, so it's coming automatically to my tongue, so I try to, yeah. So DNA, RNA, protein, they are like the, if somebody have seen any building being prepared in the near house, or any big apartment is being made or any complex is being made, you might have seen that from the bottom they make a big hole first, and they make lot of a strong foundation with lot of bricks inside that, in order to make it earthquake free, to have a strong foundation, and it could stand, it could stand the, we're gonna make 4 or 5, I don't know how many floors, but it could stand all the pressure while doing so. The same way, when we have our human body, the cells are being made, and within the cells, DNA and RNA proteins are being produced. So they are the basic building blocks of our body. So if they are strong, if our DNA is strong genetically, that what we got from our parents, only then the whole body, our whole mind, our whole working ethics will be strong. So it's all about DNA at the end. Saying said, how it is so, how it is the unit of heredity, how it could be so important is just, the reason behind it is that within this gene, within this DNA, the DNA segments carries the genetic informations in the form of genes, which was, when the Mendel was working, this our Gregor Mendel, during he was a pastor in the church, and behind his in garden he was working with peas, with a lot of experiments, doing in a lot of amounts to produce a very good statistical data. So at that time he called these factors, these genes as called as factors actually. So no one doesn't know that what he was doing, what he's working, why the purple color pea plant is producing a different color later on, why the third generation is giving different, fourth generation is giving different. So all these data was still undiscovered and doesn't know what was happening. So in that sense, but later on, some researchers came out. They work on the Drosophila, and they found that, oh wow, he discovered really a big thing. So that's how the genes, they're playing quite a big role as a unit of heredity. So it's a normal stretch of DNA. What is gene is actually? So it's normally a stretch of DNA that codes for type of proteins or RNA chain that has function in organism. How it is handling that all knowledge? This diagram is bit shabby, is not so good. So in order to make it uh, better, let me show you something else. I had one image which I shared with always at the start of the lecture, which I couldn't unfortunately find it, but I had shared in my beta fourth year class. So it might be in my earlier history. I hope I could download that. Just excuse me, student. Just because that is quite essential in order to understand the how DNA is all compiled into your DNA. I think this is not the image, or this is the image. No, this is timetable. No, it was not this one. Just excuse me, students. Just 2 minutes more. If I couldn't find it, then we stop it. Ah, because this image is not so happening. Okay, anyways, I will show it to you the next time. Let's we skip this part. In this, I can show you with here, like your this is your DNA, which is made up of your A, T, C, G. You all know these nucleotides now. So A, T, and this is C and G. They are all combined together, and this consists of DNA, which is made up of four chemical letters here, and then they get helical into the structure, and after getting helical structures, they get compiled. They get very it's like a you have a woolen no, for making your making sweaters. Our mother used to make it. So what we do, we just make the very crab, like intermingle them, make a zigzag thing, and then we make them into push, like हम इसे जोड़ देते हैं और छोटा छोटा कर देते हैं. We make it even smaller, and then we roll them around some balls here, and these balls are actually octamers and made up of histones, eight types of histones in a one ball, histone 2A, 2B, and then this complex with the DNA revolving around your nucleosome, um, around your histone is known as nucleosome. So these are your nucleosomes. Okay, and then these balls of your nucleosomes, they also get all together, make a bunch of balls, and then they got make a very bungee bunk of things together, and then they are all get settled down under your chromosomes, chromatids. So these are your sister chromatids of your chromosomes, each one of them, and these will be lying your 23 pairs of chromosomes lying within the nucleus. So that's how everything is settled down like this within the cell. So genes, so we can see these chromosomes here. So the one which is the dark part, these dark parts mainly containing your genes. So in this, gene 1 and gene 2 both are present at various levels and various things in order to work out. So first thing first is your DNA. What is DNA? It's a deoxyribonucleic acids, and why is it called deoxy? It just means the ribose sugar doesn't have the oxygen at the second carbon. That's why it's a deoxy. And it's a nucleic acids which contain the genetic instructions used in development and functioning of all living organisms and also some viruses. So it's basically the blueprint, a basic architecture which builds your RNA is built from that, and then further protein could be made. So it's always that from DNA, always RNA could be produced, then from RNA protein could be produced. It could never be that from DNA you can produce directly protein. This is not possible. No way. Only from DNA first your RNA is produced, and then your protein. Yeah, from your RNA, messenger RNA, you can also produce DNA. This is a complementary DNA with the help of your reverse transcriptase. This is one thing is possible. And then after the complementary DNA, you can produce again mRNA, and then protein could be produced. So this is also one thing possible. But this is not possible that from protein you can produce ribosome. Oh, sorry, RNA. So this is also not possible, and this thing is possible. So these are some exceptions to the central dogma of life. So this is all possible, but this is not possible. So it's so that's how things has to be taken care. Just a second. Hmm. So any question, student? I'm just teaching you very what? Keep it. Um, but I don't want these detonations. So if you have any questions so far, I have shared the image. Oh, sorry, Siddhi, it's just in the in the level of enthusiasm for teaching. I sometimes lost this, but thank you for your comment. I will take care of that from future onwards. That it should be slow. Yeah. Um, so you said you shared something on Telegram group. Aha, that's I was looking for. That's image. I just lost it somewhere. So here you can see students. Let's we focus on this image today, and then we wrap up. Yeah, till here we will make it happen. This is quite essential for some students. You know about that. For some, this might be new, but this is really essential in order to understand your genomics, in order to understand the study of or your chromosomes, which is called as cytogenetics, because this is a basic foundation that makes for our studies. So here we can see that first we have our DNA double helix at the simplest level, chromatin, which is a double stranded structure of DNA. And the diameter is around 2 nanometer. This is being asked in many MCQs in previous parts. Because this has been a bit controversial in recent years that it's not 2.9, it's 3 point something, but till now in the books it's still 2 nanometer. Then these DNA parts, as I said, they are revolved around these histones, which form in together your nucleosome. Yeah, and this is of 8 histone molecules are present. H2A, H2. I can show you that slide, like how these histones are being carried out, how the dimer tetramer is being made actually, and this is the H1A, H1 on the yellow one. It's like a clip, you know. In order to you put your clothes on the string to make it dry, so you put some clips on it, you know. So these clips really help to bind everything together. So the pairing here of your DNA, your helical DNA structure is around 1.5 or 1.6 times around it, not like 2 or 3, just 1.6 0.5 times. And then as you can see, they keep rolling, and then the diameter here, the length of your each nucleosome is around 11 nanometer. And together with the DNA and this nucleosome, it's known as your chromatosome here on the side one. And now these, your nucleosomes, your histones, covering with your DNA, they got all coiled and they produce a fiber of 30 nanometer. They produced a fiber of 30 nanometer here. Now afterwards, these fiber itself, when they were like crossing each other, you can see how they started to make the small shapes, and they also started to make fibers, and the first fiber of the length they make is around 300 nanometers, and once it is done, then it is being making even more smaller, like this crisscross Z shape of around 250 nanometer wide fibers only. And once it is done in the length of 700 nanometers, once it's done, they will make all this crisscross into this very nicely tightly coiled with 250 nanometers fibers, which produce chromatids of our chromosomes. Yeah, and total it's 1400 nanometer. Okay, so this is how the basics gene as a hereditary unit, how it functions, how everything is coiled, how it's working, you know, in a nice way. So this is very basics, very particular, and making our cell work. So thank you who shared this, Pallavi, Gaurav. Thank you so much. Thank you, Rahul, for sharing this image. Ah, wow, this is even better. Gene consists of DNA, which is made up of four chemical letters, that is your A, T, and C and G. And the chromosome here, as this we have discussed in detail now, the same diagram, how they're composed into your cells with the 23 pairs of chromosomes. Great going, great going. So yeah, so we came to an end for today's class. I hope I taught you very basics. You might be aware of, but it's just fun-loving class. Just not so serious. But we will get serious in upcoming days. The business will get serious. Things will get complicated, of course. Um, yeah, I'm just kidding. It will be not. so, so I will share the first slide of today with the cell thing. So you don't have to create assignment yet for the same. I will tell you once we are done completed with the complete day one chapter. So then you can start writing about it. Yeah. And if you have any question, Ashwin Rajiv, could you please explain it once more time? Sure, why not? Um, So basically, we have discussed how gene as a hereditary unit is being complexed into the cell in the form of chromosome. So what we have discussed here is DNA double helix structures of 2 nanometer here, just only 2 nanometers of diameter. And your chromosome on the other side is of the length of 1400 nanometers. So the journey from 2 nanometers of diameter to the chromosome of 1400 nanometers, it's all about how everything is being nicely compact and packed, and all the genetic information is available for your transcription and translation in this nice uh, little tiny chromosomes here. So what we have discussed is that in order to build these structures, we need some strong foundation. So these DNAs, they get coiled together and they get interacted with these nucleosomes, these histones, making them as the nucleosomes. And after making them, they get make a pair of fibers. These nucleosomes make fibers, and these fibers keep coiling with each within themselves, making even finer and finer and finer. At the end, these all chromosomes, including your DNA, they get bind together in these sister chromatids. So this is your two sister chromatids here, sister chromatids, and tightening calling of the 250 nanometer fiber, which produce the chromatids of the chromosomes. So in total, it's around 1400 nanometer of your length is being packed by this DNA. Yeah. So thank you very much, students. So that's it for today. No, 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 no. Chromosome is around the revolving around the part around your nucleosome. That is your chromosome. And chromosome, it's a different thing. It's the whole genetic material which is including your chromosome. That is your chromosome. So they are not the same. Yeah, you're welcome. You're welcome. So I have a group of three students want to write a review article. Need you? Yeah, you can talk to me later. I'll be happy to answer. No worries, no worries. So thank you very much. Some students might have classes later, so we will make the session end here. Thank you and take care. Yeah, and stay safe. Bye bye.